Hello, everybody. Happy, what is it? Wednesday. Happy Wednesday, if today is Wednesday for you. If not, then happy whatever other day it is for you. Welcome, everybody, to Grow the Info, where we support the proliferation of information, and I like to give you guys good sources to back up everything. That way, when you try to go and grow the info, then you have what you need to back up everything that you are trying to tell people. So, right now, we got the coronavirus scare thing going on. Um, I don't mean scare thing as in like it's not serious, because, all of the scary viruses are serious. But we are all concerned about that right now, is what I'm trying to say. There is a hearing going on with a House subcommittee on the advancement of biomedical sciences and the Department of Energy's role in that. And this hearing has been going on for probably about an hour already. I didn't realize that it was going on. So I'm going to go ahead and play it from the beginning. So, sorry it's a little bit behind, but it is what it is, and at least it will be on the channel for you guys to watch. Just so you are aware, regarding the coronavirus, there is another hearing that is happening, which will be streaming at 2 o'clock Eastern Time, 2 p.m. Eastern Time, and 1 p.m. Central Standard Time. So keep an eye out for that. Make sure that you're subscribed and turn on bell notifications so that you get notified when we go live. And I will probably be doing a coronavirus stream this evening or uploading a video. I have one recorded, but I don't like how I was coming across in it, so I might just do a live stream to just go ahead and get the information out there. So thank you guys so much for joining, and I'm going to go ahead and switch this hearing on. There aren't any white papers. Usually I like to go over the white papers and reports and stuff behind a hearing, but they don't actually have anything of that up right now. I will be adding the witness list to the description box here in a little while in case you are interested in that. Thank you guys so much for joining, and let's go ahead and get into it and get some information in our bellies.
The subcommittee will come to order, and I want to thank everyone for coming, particularly our distinguished witnesses, all of whom are exceptional and precious in their scholarship and dedication to science and to healing. We are honored by your being with us today. We're here to discuss the topic of the Department of Energy's role in advancing biomedical sciences, a transformational role at the intersection of energy and health. I'm going to repeat that, the intersection of energy and health. After all of us are composed of a complex web of neurotransmitters, pulsing energy throughout our tissues, organs, and bones every day. This subcommittee has to be about more than just what we think of as conventional power. Indeed, the department's long history and leadership in supporting basic science has resulted in biomedical innovations and a toolkit of advanced scientific capabilities that can enable future breakthroughs in health research, including artificial neurotransmitters, drug discovery, higher precision imaging to advance brain and nerve mapping, diagnostic technologies for cancer detection, and the use of technology to eliminate cancer's bad cells instead of adjacent good ones. We can think of 1,000 goals beyond those. Let us first focus on the core of human function, the brain. For over 3 decades, Congress has funded increasingly advanced research on human brain function aimed at discovering and then healing medical conditions that eluded prior generations. Now, with advancing technology and the advent of supercomputing, science itself is maturing and combining know-how, as evidenced by the wedding of biology and engineering, bioengineering, a term, frankly, that didn't exist when I went to college. And there are many more such examples in science. Our objective today is to probe where future scientific inquiry might be better directed to advance our understanding of human health, such as better comprehension of brain function, and then unravel incompletely understood brain impairments with the objective of healing. If I were to ask the question, what does medical science understand and not understand about varying conditions like bipolar, post-traumatic stress, and CTE, brain injuries, what might researchers tell us about physical properties and biologic impaired functions in each? Although the Department of Energy pursues research in areas directly pertinent to the department's main mission of energy, environmental stewardship, and national security, basic research undertaken for one purpose often has unanticipated and major impacts in other areas. That is why it is critical to support basic science research at the department, and this subcommittee surely has. In fiscal year 2020, we increased funding to the department's Office of Science by $415 million to a record level of $7 billion. This funding is foundational for a new generation of discoveries at the intersection of health and energy. For example, in conducting basic science research for other purposes, Brookhaven National Lab helped pioneer the PET scan technology that many of our colleagues have experienced in their own family life. Additionally, while pursuing its mission-driven research, the Department of Energy has developed advanced scientific capabilities that can provide unique benefits to the biomedical sciences. The department's X-ray light sources and the nanoscale science research centers provide special imaging capabilities for the biomedical research community. The X-ray light sources produce X-rays that are billions of times brighter than medical X-rays and have been used by scientists and pharmaceutical companies to develop drugs and treatment for neurological diseases like Alzheimer's, cancers, diabetes, and the development of new vaccines like the Ebola vaccine. In better understanding how drug molecules interact with human cells, hopefully, we can start to understand better why some pharmacological treatments work for some patients, but not for others. If we could more seamlessly use the scientific capacity of the Department of Energy to probe the medical unknowns, what breakthroughs could our nation achieve? The Department of Energy's high-performance computing capabilities can also provide solutions to biomedical applications through simulation and modeling, to predictive biology through artificial intelligence, and data analytics. Just imagine the computing power and data analytics needed to map billions upon billions of brain cells so we can reveal the brain's neurological and biochemical inner workings. Through the Department of Energy's Isotopes program, the national labs and a network of universities have impressive capacities in producing medical isotopes that are useful in nuclear medicine and radiotherapy. As we will hear from our witnesses, advances in the science of energy, as well as biomedicine, depend increasingly upon integrating many other disciplines with the biological sciences. It was not so long ago that universities began offering programs, as I've mentioned, like bioengineering, two sciences that had remained separated on campuses, university campuses, for generations. Given the unique scientific tools and personnel of the Department of Energy and its national labs, and the potential for future breakthroughs in health fields, it seems especially important for the Department of Energy to increase coordination and collaboration with the National Institutes of Health and other federal agencies, including, in my opinion, the Department of Defense. This will give the best chances of developing new innovations in biomedical sciences to unlock the hidden mysteries of the human brain, retool the human body when it has been broken, save lives, and sustain life on Earth. To our distinguished witnesses, we look forward to hearing from each of you. I'll now turn to our distinguished ranking member, Mr. Simpson, for opening remarks, who, as a respected dentist in his home profession, well knows the advances in technology that begin with his own profession. Mr. Simpson.
Thank you, Chairwoman Capter. Timing is everything, you know. The knee replacement wasn't bad enough. This morning I had to go and have my annual eye checkup, so my eyes are dilated and I can barely read this. So if I stumble along, I'm hopeless today. Anyway, thank you, Chairwoman Capter. I know today's topic has long been of particular interest to you, and I look forward to discussing these issues in more detail. I'd like to join you in welcoming the witnesses to today's hearing. We always appreciate it when experts such as yourselves are willing to take time out of your schedules to come and talk to us about what you do and what it is or is not working with current federal activities. Over the past few decades, we've seen many exciting and literally life-changing advances in biomedical sciences. One example I always found particularly interesting, and maybe it's because I was sitting on my patio at home during the August break in Idaho. My phone rang, and I picked it up, and it was Secretary Perry. And he was in California at Berkeley. He says, "You got to come down here and see this." What are you talking about? Secretary says, "They just showed me an example of a thing where they can take a blood sample, and within like 10 minutes they can run a thing on it. I mean, like they do when your sugar levels or whatever. They can tell if you've had a brain injury, if you've had a concussion." And he said, "Think of what that means to everything from our soldiers to people playing football. No longer do you have to go through the how many fingers is it and can pronounce it and all that kind of stuff." I mean, and that was made possible because of the computers at the Department of Energy. So there are many exciting things happening. Some of these advances have come about due to, at least in part, to the Department of Energy, whether it's research being done at the DOE user facilities, such as the light sources, or research intended to advance DOE's missions that was found to have biomedical applications. DOE has been an important partner in advancing biomedical research. I look forward to hearing more details on how DOE can improve its partnerships with NIH and relevant non-federal entities to make even better use of the user facilities and research efforts to further advance biomedical science. I'll also like to hear how we can improve these partnerships in a way that maintains appropriate focus on the fundamental missions of the separate federal agencies. Advances in biomedical science are certainly in the national interest, but so too are advances in energy, environment, and national security. In fact, I'd offer this cautionary note. Today, NIH funding in fiscal year 2020 was $41.7 billion. Funding at DOE's Office of Science, where most biomedical-related work is done, was $7 billion. So we need to ensure that improving collaboration between these agencies does not lead to diverting the already much smaller DOE science budget away from advancing DOE's missions. Better collaboration between DOE and NIH can be a win-win for both agencies and both missions. We just need to make sure that we keep that goal in mind as we look at developing future efforts. I look forward to learning from our witnesses today about the challenges, the successes, and the ideas for improvement in this area, and I thank Chairwoman Capter for calling this hearing.
Thank you, Mr. Simpson, very much. And obviously, we're very excited for our witnesses and to hear from them. I just wanted to add a sentence. What partly tripwired this hearing this morning was I was out at Walter Reed's Intrepid Center. And in meeting with our veterans and looking at some of the conditions that they are enduring. And we walked in one room there, and I saw a machine I'd never seen in my life, called an electro machine invented by the Swedes. And I watched a veteran go through the imaging, and then up on a screen, very quickly, they were able to go deep into the brain and identify the filament that had caused his most recent stroke. I was completely fascinated. I felt like I was in a new age. I thought, I'm really in the 21st century. I'm really, okay. So how do we, knowing what we do here, how do we help the science advance more quickly and go deeper into the human body and help healing. So our witnesses today are extraordinary. First, we will have Dr. Harold Varmus, who is the Lewis Thomas University professor at Weill Cornell Medicine and a recipient of the Nobel Prize for studies of the genetic basis of cancer. He served as the director of the National Cancer Institute for 5 years and director of the National Institutes of Health for 6. Dr. Varmus also co-chaired the Secretary of Energy's Advisory Board Task Force on Biomedical Sciences. These top-level scientists were looking forward even then. Following that, we will have Dr. Mark Chance, the director of the Center for Proteomics and Bioinformatics at Case Western Reserve University in the state that I represent, and vice dean for research at its School of Medicine. Dr. Chance also directs the Case Center for Synchrotron Biosciences located at Brookhaven's National Labs Light Source. And I can recall a moment when the director of that lab gave me a lesson on ions, for which I was very grateful and remain so. And finally, we will have Dr. Narayanam Kasturi, a neuroscience researcher at Argonne National Lab, who is also an assistant professor of neurobiology. That name didn't exist when I was in college, at the University of Chicago. Thank you all for taking the time to be here today. I want to thank our members for attending. Without objection, your written statements will be entered into the record. Please feel free to summarize your remarks, although I want to hear every word, and starting with Dr. Varmus. Welcome.
Well, thank you. I think I was audible before. Let me start with a brief reminder. The body, as the chairwoman has pointed out, is made up of physical entities, and the strengths of the Department of Energy have been reflected in medical science and responsible for many of the advances we've made in health for a century. Think about imaging with X-rays and radioisotopes and MRI imaging. Think about the way we monitor the heart and the brain with electrocardiograms and electroencephalograms. Think about how we analyze the blood by looking at chemicals. Think about how we take all that data and crunch it with computational tools, and how we simulate with engineering tools. These are just a small sampling of the disciplines that have been championed by the DOE Office of Science, physical sciences, engineering, computation, that contribute to biology and medicine. Now, as the chairwoman has pointed out, I've had a number of federal positions in which I've been able to observe some of the contributions made by DOE from a front row seat. Let me just mention two. First, the Human Genome Project, if we can have the first image, please, which was initiated at a DOE lab in the 1980s. Most of the genome of human beings and other organisms were determined by a small number of centers, one of which was run by the DOE. Even though the NIH was coordinating this international project. And these share interests persist through computation, the study of many other organisms, and through the development of faster and cheaper machines for sequencing DNA. A second example is in structural biology. And when I was director of NIH and Ernie Moniz was the Undersecretary of Energy in the Clinton administration, we made a deal that actually transferred money, Mr. Simpson's point, from the NIH to DOE synchrotrons to sustain the beamlines for doing structural examinations of important proteins that serve as machines for carrying out cell function, the ribosome for protein synthesis, complex enzymes to copy DNA into RNA, membrane proteins that react with drugs are responsible for the transport of water across membranes. And these interests also persist through the development of cryo-electron microscopy, neutron beam, for pursuing the structure of proteins at a more refined level. Now, as the chairwoman alluded to, after I left the National Cancer Institute in 2015, sorry, I'm not that old, in 2015, Ernie Moniz, who was then the Secretary of Energy, asked me to join the Secretary of Energy's advisory group and to co-chair a working group on the topic of today's hearing. And we were asked to assess the opportunities for better coordination between the NIH and the DOE's Office of Science and to strengthen those relationships. We conducted several workshops to talk about a variety of disciplines that are particularly relevant to DOE's activities: material science, fabrication, nanotechnology, sensor development, radiobiology, imaging, advanced computation, other things, in the context of many of the existing initiatives then at the NIH, in precision medicine, the cancer moonshot, the so-called BRAIN program, microbial drug resistance, and others. And in the course of these hearings, we learned something quite important about the difference in the scientific cultures supported by these two agencies. The NIH traditionally depending on the imagination of individual investigators with small labs getting individual grants from the NIH, the DOE through its national laboratories carrying out mission-driven science, developing technologies, and that fundamental cultural difference is an important one I'll come back to. We recommended many ways in which these past activities could be enriched and fostered, but we came out in addition with 4 overarching recommendations that are relevant to today's discussion. How we convene panels of experts to identify opportunities for research programs, how we share the cultures of the two agencies, how we establish facilities for large-scale, newly identified missions, and how we can report more often, as we are doing today, to committees like this about what we do. I'm going to just say a few words briefly about the consequences of this report that we wrote for CAB. Because I've found in response to the invitation to speak here today by talking to many officials in both agencies that the spirit and the letter of the report are thriving through joint projects, large and frequent consultations, and work of individual scientists engaged with both agencies. I'm going to mention, in conclusion, if I can have the liberty of one additional moment, Madam Chairwoman, 2 joint programs that are important to me. One is a current initiative in the cancer domain, which is my own area of research. And as you can see, maybe you can't see because the type is not that large, there are a series of the many things going on between NIH and DOE, a set of projects that involve the National Cancer Institute's mission to study better ways to improve outcomes for cancer patients, mainly through making better use of high-performance computing. Second, to be able to visualize in higher resolution the position and function of proteins that we know to be responsible for driving about 1/3 of human cancers, and another pilot project that helps to enrich our epidemiological surveys. The second of these is actually being carried out at the Frederick National Lab for Cancer Research, a national lab that is run by the National Cancer Institute. When I was director there, we modeled that lab carefully on the examples provided by many of DOE's national lab, establishing an initiative to study what we call RAS proteins, these proteins that drive cancers. And we now have, if we can move the slides along, an effort that is bearing fruit to study how RAS proteins interact with cell membranes in ways that I don't really have time to describe in detail. That model has been expanded to include public-private partnerships. If you can advance the slides too, one more, to include not just DOE and NIH, but University of California, San Francisco, the academic sector, and the private sector. I'd like to conclude with a suggestion. In our report, we talk about scanning the horizon for problems that we could solve through national labs and interactions between NIH and DOE. You may have seen a number of reports that talk about the way in which the pharmaceutical industry and the biotech industry are having difficulty financially and scientifically in coping with the spread of organisms that are resistant to antibiotics. Many of these commercial forms are retreating from the foray to try to identify new antibiotics because the economics don't favor that kind of investment. And there have been stories in the New York Times and elsewhere on this topic. I think that it's possible to consider, next slide please, taking an organization like that resembles the Molecular Foundry at Berkeley Lawrence Lab and turn that into a place where individual NIH-sponsored students of new antibiotics and microbial threats could be put into a setting where the flexible and deep technological advances could be used to identify new targets for antibiotic action, to try to find new antibiotic molecules, and use that government resource and the collaboration between these two agencies to meet a problem which WHO and NIH and many others see as one of the great threats to human survival in the next century. So I want to thank you for having this hearing. I want to thank the two organizations, NIH and DOE, for their activities in the past to sponsor these highly active and fruitful collaborative arrangements, and be pleased to answer any questions you might have. Thank you for the extra time.
Thank you, Dr. Varmus. Thank you for your time. Dr. Chance.
Good morning. Chairwoman Capter, Ranking Member Simpson, thank you so much for the opportunity to talk today. My testimony, I hope, is very simple. DOE science facilities are critical to America's basic biomedical science, drug and device development, and public health goals of the nation, with positive effects on the health and welfare of all Americans and saving many lives. What's my evidence? Well, I'd like to focus on new drug development, where over 90% of the newly approved drugs result from projects that use DOE facilities, specifically synchrotron methods, to see the structure of the drug's target. This site allowed the drugs to be developed more quickly, as well as resulting in safer and more effective medicines. In the first slide, we can dig in a little and explore these DOE facilities called synchrotrons. There are 5 of them across the country. They're large physics facilities producing very intense X-rays for a wide range of scientific applications: materials science, chemistry, physics, and biology. With the help of Brookhaven Labs, we did some research for this hearing and found there are 42 dedicated biomedical beamlines currently at the synchrotrons around the country. 3,000 biomedical, or about 4,000 biomedical science users among the 12,000 users overall of the synchrotrons. Dr. Varmus alluded to the Nobel Prizes in biology, and the users published 1,200 papers last year and enabled 100 companies to move their research forward. If we could go to the next slide. I want to introduce the Center for Synchrotron Biosciences, as Chairwoman Capter noted, is located at Brookhaven National Laboratories. I established the center many years ago to make collaboration between the NIH and its funded users, academia, and the DOE easier, to provide a platform to develop collaborations, to bring in technologies, and most of all, in terms of that cultural piece that Dr. Varmus referred to invent new uses of X-rays and other light beams to see the structures of drug targets and other biomolecules in novel ways to speed medications to patients. The results have been very gratifying for myself and my colleagues. Over the years, we've built 10 beamline instruments. Hundreds of institutions have participated, thousands of users, 2,500 publications so far. The funding to build and operate these facilities has come from the NIH and the NSF, and we've made key technology partnerships with various organizations to move things forward. One of our most recent partners is the NIH and the Cancer Institute and the RAS program, which Dr. Varmus referred to. If I could have the next slide. So one of the things we did, and Dr. Varmus referred to it, is we stepped up the rate of structure determination. We partnered between the NIH and the DOE, and we built beamlines that could allow us to see these drug targets, many more of them. And I think the results are pretty good. There were fewer than 10,000 protein structures before we started this collaboration. There are 160,000 now. And they're coming out at a phenomenal rate. And these structures were determined at DOE synchrotrons and also around the world, where these synchrotrons are considered very important. Just to add, there are probably 100 synchrotrons under construction or in operation around the world right now. So what's the data? Well, if we looked at the 210 new drugs that were approved between 2010 and 2016, 93% of them relied on protein structure information provided by DOE and other synchrotrons. That was about a rate of 30 per year. We're now, the last 3 years, we're 50 a year. And you know why that is, at least to me, or what much might be plausible? There's a delay between the time you finish the structure and you get the FDA approval. And you see that curve? 10 years ago was on a very steeply rising part of that curve. So I would argue we're on the cusp as we bring in machine learning, artificial intelligence, and the computational power of the DOE to accelerate this process dramatically. If I could go to the last slide. What's coming? New facilities, new beamlines to solve new challenges. And the Center for Synchrotron Biosciences is in the forefront. We've completed a new beamline at Brookhaven to solve important medical challenges. The number one we've just solved is we've solved the structure of the human nuclear receptor. That's going to be important for tamoxifen-resistant breast cancer, which develops. Nobody knew what that molecule looked like before, and it was really hard to drug, and it was really hard to approach without seeing, because seeing means understanding. We're also solving structures of proteins important in Alzheimer's and mad cow disease, and very important to today, we're studying virus structures. Just last week, Purdue scientists were at the Argonne synchrotron looking at forms of coronavirus that matched the Wuhan strain to try to rush vaccines and drugs into development. So again, I thank you so much for the opportunity to speak today. In conclusion, I want to thank you, the appropriators. Your investments are paying off, and thousands of Americans' lives are going to be changed as a result of it. I'd be happy to answer any questions. Oh, do we have one more? Do we have one more slide? Do we have the shoe?
Oh,
if
please
with
proceed,
your
Dr.
permission,
Chance, yes.
Madam Chairwoman? So we were thinking to try to illustrate to the committee the scale of these facilities. They're expensive, right? So I thought, let's give an image which many in Ohio know. That's Ohio Stadium, the shoe. And I had a side bet that the National Synchrotron Light Source 2 was bigger than the shoe. And as you see, a half one, right? The shoe's taller, it's on top, and there's the National Synchrotron Light Source. NSLS is bigger, so the bet was a push. Nobody won. But thousands of people, 100,000 people are entertained on a Saturday in the shoe, and thousands of the nation's researchers are served yearly at the National Synchrotron Light Source 2. Thank you.
Thank you, Dr. Chance, very much. Dr. Kashuri.
Chairwoman Kaptur, Ranking Member Simpson, members of the subcommittee, thank you for the opportunity to appear before you to talk today. My name is Bobby Kasturi, and I'm a neuroscientist in the U.S. Department of Energy's Argonne National Laboratory and an assistant professor in the Department of Neurobiology at the University of Chicago. And what I'd like to talk to you about today is that we stand at a pivotal moment in our centuries-long quest to understand the brain. It's actually a moment when multiple scientific worlds are colliding. The worlds of computer science, bioimaging, and microscopy and neuroscience are now colliding. My research specifically leverages the capabilities of the National Lab System to accelerate that collision, specifically by mapping brains at the finest scales and analyzing the resulting enormous data sets. It is from this perspective that I present to you my testimony today. As you heard, one of the great intellectual challenges of the 20th century was our understanding of the genetic basis of life, and the Human Genome Project is perhaps the pinnacle of that effort. It's important to remember that the Human Genome Project was born out of the collaborative efforts of the National Institute of Health and the DOE. It's perhaps one of the first and most successful big data and healthcare collaborations ever, and its impact continues to reverberate through biology and medicine. It is also, for the purposes of this hearing, an example of how the DOE and the national labs are uniquely suited to play a similar role in tackling the great intellectual challenge of the 21st century, which is mapping and understanding the human brain. And it is the enormity of the human brain that requires this collaboration. The human brain, your brains, contain about 100 billion brain cells, or neurons, each of which makes on order 10,000 connections with each other. One way to think about that is a single brain contains more connections than there are stars in the Milky Way galaxy. And the complexity of this intricate communication web cannot be overstated. As neuroscientists, we believe that making a map of these connections could provide answers to some of the fundamental questions about how brains work. Such a map, which we call a connectome, is a map of everything about you: your skills, your memories, your fears, and your personality. Disruptions or alterations in this map, miswirings, if you will, between neurons, are the basis of many neurological and psychiatric disorders. Using facilities like Argonne's advanced photon source and high-performance computers, we plan to map the normal brain, which will allow us to treat diseases like autism spectrum disorder, schizophrenia, concussion-related neuropathies, and dementia. To kind of give you an idea of what is capable, if we could play the first video, please. What you're looking at is, based on work I started as a postdoc, is the first map of a human brain connectome by teams at Harvard University and Google. You are seeing the individual processes and connections of millions of neurons in the human brain, traced by AI algorithms developed by Google. And for the first time, you will see three-dimensional models of connections in the human brain. Each of those is one of the many neurons in the human brain. But even this effort, which tests the resources of bleeding-edge researchers, represents much less than 1% of the volume of the entire human brain. The raw data of the Atlas of Human Connections, i.e. the human connectome, would require approximately 1 trillion gigabytes, a zettabyte, and could not fit in the memory of any computer today. The entire Human Genome Project requires maybe a few gigabytes. Another way to say this is that all the written material in the Library of Congress, all the movies and audio that humans have produced since the dawn of civilization, would be just a small fraction of the data of one human connectome. Thus, the interdisciplinary capabilities of the DOE national laboratories and their world-class user facilities like computing are our best chance to transform the way we understand the brain. Scientists from Argonne National Lab and the University of Chicago, in collaboration with researchers across the United States, including Case Western, are already working towards a human connectome by mapping smaller brains of other animals, such as mice. But for most neuroscientists, the tools that require to map the brain, particularly the computing, are too expensive and require investments in physics, engineering, and computer science, not only beyond the scope of individual laboratories, but probably entire universities. These barriers prevent the neuroscience community from asking the best questions about the brain and limit the type of collaborations that drive innovation. We need to democratize this widening gap between the small fraction of laboratories developing and utilizing the most advanced technology and the majority of neuroscientists. A collaboration between DOE and NIH could provide these maps and the tools to make these maps to the national neuroscience community. A revolution awaits us when we understand how human brains acquire knowledge from experience, how we find patterns in our senses, and how we plan things and make decisions and act. When we know exactly how these processes work, we can connect prosthetic limbs to the paralyzed, design rational medical treatments for brain diseases, and ultimately try to reverse engineer human cognition into our own computers. As I said, we stand at a pivotal moment in the history of our country. Understanding the brain will help provide U.S. leadership in neuroscience in the coming decades, the way the moon landing in 1969 and the Human Genome Project contributed to U.S. technological and scientific leadership. The national laboratories, the source of some of our most powerful scientific tools, combined with the research and development of the NIH, can revolutionize our understanding of the brain to develop better treatments for millions of people worldwide affected by neurological disorders. And perhaps most importantly, the DOE and NIH together can achieve generational science, removing boundaries between fields of science and creating these transformative moments that shape society for generations to come. Thank you for your time and attention today. I welcome any questions you have.
Thank you very much, Dr. Castori, and all of our witnesses. We appreciate your statements, and they will be placed in the record along with any other materials you wish to submit to us. Since this is the first hearing of the year, I'd like to remind members about our hearing rules. For those members present in the room when we gavelled in the beginning of the hearing, I will recognize you for questions in order of seniority, alternating between majority and minority until all who arrive prior to the gavel have asked questions. For those who arrive after the hearing has started, I will recognize those members solely in order of arrival, and again, alternating between majority and minority. Lastly, I intend to try to observe the 5-minute rule for questions and answers, and we will now begin questioning under our normal rules. Dr. Castori, I wanted to begin with you. One of my own deep interests is understanding the function and impaired function of the human brain to advance diagnosis and treatment for those who suffer from neurological and biochemical brain imbalances. If one looks back at the last 5 years in our nation, we have witnessed some troubling trends. The number of individuals, particularly young men between the ages of 15 and 35, who are taking their own lives. As well, we know we have large numbers of afflicted warriors, wounded warriors experiencing brain trauma and post-traumatic stress. We have an opioid crisis with tens of thousands of harmful self-medicators, and there are harm perpetrators in our society taking the lives of others at a very troubling rate. Most have needed brain-centered medical help, but it was either not available locally, nor if diagnosis was accurate, a proper healing course of treatment not able to be effective. Because we're members of the House, we experience these individuals at ground level. And it becomes part of our responsibility to meet that human need. I believe improved diagnosis of neurochemically brain-centered medical conditions to be a significant national priority. Medical science needs even better imaging as part of the solution. One of the first types of brain scan or neuroimaging occurred in 1927. It was called the cerebral arteriogram. You all know this. But it took about 3 decades later until computerized tomography emerged to benefit medical diagnostics. A little later, magnetic resonance imaging was developed by researchers. I can still remember as a young member of the Veterans Committee when I first arrived, when at one of our VA centers, they turned it on and anything that was metal attached to it. And the nurses, were running down the hallways, so we witnessed this in our own lifetime. This technology identifies atoms by how they behave in a magnetic field, a highly useful, non-invasive procedure for imaging internal bodily structure and advancing medical diagnostics well beyond where the technology had previously existed. Existing imaging technologies have transformed medicine forever. But we ask ourselves, what are the next horizons? I mentioned my experience at Walter Reed Intrepid Center, where the researchers and doctors are trying to help veterans who present with a whole range of conditions based on their military service and having been wounded in theater. Dr. Castori, what can we do to advance technological research faster, to improve imaging technologies or other technologies you would all recommend, to better capture deep brain tissue and neurochemical function, and to identify malfunction? How can we better brain image more precisely to map neurochemical dysfunction that may be occurring, as well as understanding reactions to pharmacological or other treatments to improve patient outcomes? I know that's a mouthful, but if you were to sort of point the ship in the right direction, what can you add to your formal testimony to guide us?
Chairwoman Capter, thank you. That's an excellent question. I'll try my best to give my version of it. Many of the things you mentioned, I'm not an expert in personally. Some of the other techniques, I work specifically on the very finest resolution reconstructions of the brain at the level of how every neuron connects to every other neuron. Since as neuroscientists, we know that that is likely the basis of both our normal behavior and our aberrant behavior, getting those maps will be the first steps to sort of understanding how the physical circuitry underlies these behavioral defects. So I think there's a lot of technological advancement that we can make. But I think the second point that I'd like to emphasize is that there is a lot of technology already currently available. A lot of technology is being developed thanks to funding from committees like you and other committees for the Brain Initiative, et cetera. I think a real problem that we could solve right away is giving access to that technology to the broadest set of scientists possible. And I think that this is a place where DOE user facilities combined with neuroscience questions can have immediate impacts on distributing technology that's already available. In terms of how do we get to future technology, I think the answer is more money will always help for the future. But for now, I think there's an opportunity to take the technology we already have and distribute it as widely as possible.
Thank you. Would any other witness like to comment on that question? Dr. Varmus?
I think there's a useful analogy here to the...
Could you please turn the microphone on, Doctor?
There's a useful analogy to the Human Genome Project, which we undertook...
Turn it on again.
Sorry. Now I have the red light going. Thank you for your technological assistance. There's a useful analogy here with the Human Genome Project, where we began the project not knowing whether it was technically feasible, but we had a pretty good idea of how we would use the data once we got it. Because we knew that genes encode proteins, proteins carry out certain functions. I think in the Brain Initiative, we're taking a more adventurous step, and one that I think is going to have remarkable dividends, but it's one we undertake not knowing what we'll make of the connectome once we describe it. So the technology is developing. It's very important to do this, but we have to do this recognizing that we're taking on a remarkable, revolutionary adventure, one that's incredibly important to the survival of our species and one that's going to have tremendous dividends, but we have to undertake it recognizing that simply having the connectome is not going to tell us how these diseases arise, and we're in this for a very long haul, and I hope we'll be there.
Are we properly organized in terms of the connectome to really advance the science quickly?
So it's hard for me to are we properly organized at a national scale? My suspicion is no, because it's so recent. It's such a new technology and a new capability that I think we have an opportunity to organize at this moment. And I think it's the right moment to think about national organization of these efforts. I think Dr. Varmus would know how to organize at a national scale, and I'll let him.
Well, I would just point out again an analogy with the Genome Project. That began by looking at the genome not of human beings, but the genome of bacteria, of worms, of flies, and indeed part of the connectome of the fruit fly, the traditional model organism for experimental biology, has just been published. And I think we can, by observing what happens with those preliminary incomplete connectomes in model organisms, we'll learn what's needed to bring the right people together around the table to try to interpret, just as we brought people together from a variety of medical fields to interpret some of the early genomes that were deciphered. So I think there are models here to look at to try to figure out the best way to organize the right people to deal with these tremendous data sets. But DOE should certainly be among those folks involved.
Well, we obviously would benefit greatly by your thinking through how best to organize that inquiry and, what DOE's role might be, and where that might be housed or which places it might be. It already is, in a sense, but what we might do to help you advance knowledge there.
It's a question worth posing to some of the panels that are currently meeting between DOE and NIH to talk about how the two organizations work together in other domains where the rules are perhaps better understood. But I think you already have people who are sitting down on a very regular basis to talk about what brain research has been done, about genomic research, about cancer research, about research on antimicrobial organisms, antimicrobial drugs. I think there are places where you could get very good information about how to organize the larger effort to understand these very rich data sets that will be coming from research of the kind Dr. Casteri was describing.
Where was the fruit fly connectome? Where was that done?
It was done mainly organized by Howard Hughes Medical Institute at Genelia Farm and in collaboration with NIH-sponsored investigators. I'm sure that by consulting with people from both agencies that are working within the domain of the BRAIN initiative, you can identify the right people.
Dr. Chance?
I'd just like to add one point. I agree completely with my colleagues, and I just want to emphasize why it's important. Because in mental health diagnoses or pain, we don't generally have independent ways of seeing if you're getting better. We ask you. And it's just not that precise. But if we could have a meter, right, that was unbiased and quantitative for some of these things, then we can really see if the pills or the behavioral interventions or the other things we're doing are really making a difference for the patient. We lack that unbiased measure as opposed to how you feeling today.
I have to put this on the record because I've worked on it for almost 20 years. And this relates to returning veterans who have all sorts of conditions. And I'll just take my own state. We worked with our National Guard and actually Case Western to collect, along with several other universities around the country, working with our National Guard, specimens, DNA specimens voluntarily given from veterans. So to try to contribute to a bank of DNA that one could then study, why did the veteran have this condition versus that condition? And we worked with scientists at Case, and why would someone develop PTS and someone else not, all right? But as the scientists worked, their conclusions had nothing to do with the brain directly with what we're talking about today, but rather that you would develop a condition because you had experienced violence prior to enlistment in the military, and that wound remains, and then if you get in a situation where there are other stressors that happen, you develop it faster than someone else. I was very gratified for the conclusion they came up with, but I say to myself, we have this mammoth set of DNA samples. Where does this fit in? Where does the diagnostics for this fit in? And I just point that out to you, Dr. Chance, because if you go visit the Walter Reed Intrepid Center, you will see what we're trying to do there to help our returning veterans. And I know this isn't the Veterans Committee, but the machines that are used and the technologies that we're engaged with, some of them came out of the field of energy. We really have a very poor understanding of what is going on. And so my question really is, if you could go back to the case and take a look at who has these DNA, the DNA bank that has been developed, and does it fit anywhere in medical research? They've certainly attempted to work with the Department of Defense on this, and but they're not the places that are inventing necessarily the kind of technologies that you are. But for a member, it's frustrating to have this rich bank, but underutilized in terms of research. So I just point that out. Maybe other members have other experiences, and I will turn this over to Mr. Simpson now for his questioning. But it just seems to me to be hard to get your hands around, okay, what can we do to move this field faster and to get better diagnostics? Thank you. Mr. Simpson.
Thank you, Chairwoman Capter. Dr. Chance, I'm shocked that after having just had my knee replaced and going into the doctor, that when they say, what pain level are you at today, and they have those 10 faces, that's not a scientific analysis of my pain level.
That's about as scientific as we've got today.
Yeah, that's about right. I sit there, they say, what pain level are you at? And I kind of go, I like that guy.
So you see how hard it is to really know if our interventions are working, because
Yeah.
it's based on how you're feeling, that all of the things that are integrating into you as a person.
Yeah.
And maybe it's just the knee we care about for
Yeah,
this
that's
visit,
right. Yeah. Anyway, thank you for coming today, all of you, and for your testimony. What you do is vitally important to this country and to the people of this country. I have always been a fan of NIH. I served on the Labor H Committee, whenever I get depressed, it seems more frequently lately, but no, whenever I get depressed, I go out to NIH, because it is fascinating. I always walk away from there amazed. I do the same thing when I go to our 17 national laboratories and the work that's being done at those laboratories. I walk away, I don't understand any of it, but I know it's amazing. But we're an appropriation committee. And the question I have is, if you go back to, could you go back to Dr. Chance's first slide? Just out of curiosity. Yeah, you just had it. That one. I need it longer than that. Okay, you talked about the impact of this DOE light sources on biomedical research. Who pays for that? What is our role as an appropriation committee with the Department of Energy? What do we pay for? The drug companies that get the benefit of that research and stuff, how much do they pay for? How do we, I'm trying to figure out how I could determine what an appropriate level of appropriation is and what I'm going to get out of that appropriation without interfering with the role of what those laboratories are. And I am one who believes that collaboration and using things to the best of their best uses is vitally important. But do you understand my question?
Yes.
I'm not sure I do. But
yeah. There's many stakeholders who come to the table and have paid for those beamlines. There's some money came from DOE. There's some money came from the National Institutes of Health. There's the money from the National Science Foundation, which was used to build the beamline I operate currently. And the universities are putting in money. The companies are putting in money. Foundations are putting in money. So it's quite shared. And so those 42 beamlines, you didn't have to pay for all of them, right? That is, you put up you put some table stakes up, right? And then everybody said, yeah, I'm in. This is consistent with my mission. I want to play. I want to be involved. And so that's how we attract investment to your sort of base support you provide for the DOE. So as a university researcher, I'm out looking for collaborators, grants, so I can bring money, ideas, and partners to the facility to get the job done. So what's the right mix? That's a very hard question. And we have issues of, agencies have missions, and OMB doesn't like lane jumping, I've heard.
yeah,
Right?
believe it or not, yeah.
Yeah, believe it or not. So how do we sort of be intelligent about that to encourage collaboration with that? And we don't want lane jumping either, right? You don't want to be paying 2 or 3 times for the same thing.
Right.
So how do we do it efficiently? And by enabling, the different stakeholders to get, I think Dr. Varmus laid out the steps, right? The 4, we need to convene, we need to understand cultural differences, we need to make sure the facilities are operating at peak technology and in terms of numbers, and we need to tell you about it and get your feedback.
Comment briefly upon this, because as I alluded to in my testimony, in the 1990s, when Ernest Moniz was the undersecretary, we worked out an agreement by which certain NIH institutes, based on the grants they were giving to structural biologists who are working at Brookhaven and other national labs, we made an agreement that we would pay for a certain fraction of the total cost for maintenance of the synchrotrons that were producing those beamlines. And that's continued over the years, and being with adjustments to according to use. I think we actually have made, by bringing in other potential funders, particularly good use of the infrastructure initially provided mostly by DOE with supplementation from NSF, as Dr. Chance has pointed out. I think that's been quite well managed, but it would be useful for you to bring in some people who are responsible for those allocations and check out whether they're in accord with your principles as appropriators.
I'd like to make 2 additional quick points, and one is National Science Foundation. What are they doing developing drugs? Well, they recognize the important basic biomedical science that would be enabled by these beamlines. And they're okay if other good stuff happens. The second thing is the industry. Is industry paying their fair share? So DOE thinks a lot about this. And they call them user facilities for a reason. Everybody should have a chance to be a user. But if you're willing to publish in the open literature and make all your results available, we'll give you beam time based on an evaluation of your technical expertise and project goals, peer review. If you're a company and want to develop and patent some drugs, DOE calculates the full cost recovery and charges you for it in advance. I know that because I'm an owner of a biotech company in my outside interests, and we use the synchrotron, and they're very picky about making sure our funds clear before the beam time starts, okay? and they do work very hard to make sure that we're allocating the resources, appropriately that way, and I laud them for that.
Thank you. I just wanted to respond to one part of your question, Mr. Simpson, which is what do you get out of it, or what do appropriators get out of it? And we've had a lot of good slides about the immediate effects and numbers, but I'd like to add one more thing, which is that a lot of these big science investments or big science projects wind up inspiring people in ways that it's very hard, that's real, but very hard to calculate. And the way I tell the story is that in 1969, 50 years ago, and a few months from now, from today, when we landed on the moon, the average age of a NASA scientist was about 29 years old. I'm not sure what the average age is now, but it's older than 29 years old. And that number sticks in my head because in 1962, when President Kennedy announces that we're going to the moon at Rice University, many of those NASA engineers are 22 years old. They're in college or just out of college. And something about this idea of a national effort inspires them. And I think there is an argument to be made why was the moonshot a success? When I was young, I looked forward to moon bases when I was an adult and traveling to the moon for holidays, none of which happened. So in one sense, you can imagine that the moonshot was a failure. But in another sense, I suspect that the reason we have satellite technology and telecommunications and many of the things that we've had now is because we inspired a generation of students to enter space. And I think when you think about appropriations, this is something worth thinking about, that there is inspirational value that has real benefits to this country that's hard to quantify, but I would argue is very real.
Thank you for that. I appreciate that. I agree with you. It is, I guess one of the challenging things is, as you know, there's never enough money. We've got an appropriation and there's viable needs that need to be appropriated. And what I'm trying to figure out is how do I determine whether we got $50 million here, where's our best investment? And how do I determine what we're getting for that investment? And that's the challenge I think this committee has. But I do appreciate everything you do and look forward to working with you on these. You're back.
Thank you, Mr. Simpson. You know, when you look at the additional funds that have been given to the department, been passed by Congress and signed by the President for this year of fiscal year 2020, and you think about the fields in which you have devoted your lives, trying to communicate to the outside world about a moonshot type effort. I think in this field, it kind of gets lost because we heard about, even I as a member of Congress, I remember when we had members from Massachusetts who fought so very hard for Sil Conti being one of them many, many years ago for the original work that we did on the brain and brain initiative. And then it kind of, after the first announcement, it kind of goes out there and the road markers aren't clarified as it moves forward. Then, you know, you have the Human Genome Project and you hear about that and sort of, so I think in this arena, whatever it's entitled, I think it needs better road signs as we move forward to the extent one can so that people continue to be engaged, Doctor. But even myself, I know how frustrating it is for me trying to express this to an audience. It's a little murky. And we know that as science is developing, obviously it would be. But then people lose focus. So I just would encourage you to think about that and how one would organize going down this road collaboratively among interested departments and agencies. I think the public would welcome it, but I don't think from a messaging standpoint, we necessarily do the best job. So just wanted to put that on the table. Congressman Pocan.
Thank you, Madam Chair, and thank you to the panel very much for being here. Very interesting. Let me try a couple areas if I can get it in my 5 minutes, but one in particular, Dr. Varmus, you mentioned isotopes. In my district, we have a couple companies that were fortunate to get some partnership with the Department of Energy to manufacture the isotope Moly-99. And we weren't as successful in getting as much funding for domestic production as we'd have liked to in the last one. We're hoping to try to make that case this year, and I thought maybe you could help me make the case, because as we understand the domestic production where this is currently produced has been dwindling. I think the last area was I think it was in Canada on this continent, and that's why they put the grants out a number of years ago. A couple of these now have had successes. Can you just talk a little bit about how important it is for that funding to find the domestic source to be able to produce something like this, and what that means other than just a company producing it, but why we need to have domestic production of this type of an isotope?
Well, I have to do something that I don't like to do, which is to admit my ignorance about this particular substance. In general, I do think it is good for us to be producing our reagents in this country. It makes access easier and cheaper. But and on this particular isotope, I really can't comment. Maybe my colleagues know something about this.
I don't. Is this a diagnostic reagent, molybdenum-99 for imaging?
Yeah. Yes, exactly.
Yes. Well, these isotopes, it's a tough market for a small company, right? You got a few. So I don't know. The DOE is very good at enabling technology development and companies, but we need these isotopes to see the stuff we need to see. And each one has sort of a different niche of imaging that uniquely provides important capabilities. And I'm not, your folks at Minnesota and others and Wisconsin are using these isotopes a lot, and there are tremendous research centers there.
Thank you. Let me go to brain as well. I think when you mention a few things really hit things I hear in the district. People concerned about autism. You mentioned autism, Alzheimer's, depression, and mental illness. When you talk about other benefits, something like at least 30% of the people who are incarcerated are there because they have mental illness, right? Those three areas, can you just talk about some of what we're seeing in developments that might be promising in autism, Alzheimer's, and depression and mental illness?
So broadly, I think in neuroscience, there's a lot of progress being made in these fields. Some of it is trying to figure out, are there predispositions in your genes or et cetera that allow you to get these diseases more easily than other people? That's the first. I think second, there's a lot of progress in being made about how we diagnose or define these diseases, because many of these diseases aren't exactly one particular disease. They're a spectrum of diseases, and the better we understand where a patient falls on that spectrum, the better we can treat them. I think these are broadly happening. It's not the work that we do. The work that I'm particularly interested in is that for many of these diseases, for example, autism, it's this idea that it's an inappropriate wiring between neurons. Maybe those neurons are making too many connections with each other, not enough connections. And that is the fundamental basis of the disease. And so while these other advances are happening in the therapy and how we define it and how we diagnose it, we're interested in giving the sort of deep fundamental physical basis. And once we can collect enough data in that world, are there new therapies that we hadn't thought of? Because now that we understand the biology at some physical level, and that's what we're hoping for as well.
That's true with depression too, right? The connections, because I know that another company in our district got some funding recently for psilocybin research, actually trying to do new connections for people with depression, and
Yes.
it just seems to keep resurfacing in a lot of our conversations.
Yes, and I think it will keep resurfacing until we can have a definitive answer on it. And I think the interesting thing about definitive answers is that some things no longer surface that we used to think were relevant, and new things now surface that we never thought of.
Gotcha. Great. No, I appreciate, again, we just have with the University of Wisconsin, clearly a lot of offshoots working in all these areas. And, we are fortunate to get a lot of funding with the Office of Science as well, which we want to make sure that continues. And really appreciate your time today. Yield back.
Thank you, Congressman Pocan. Congressman Fleischman.
Thank you, Madam Chair. Madam Chair, I want to thank you and the ranking member for having this hearing today. It's educational, it's insightful, and it's optimistic, and I appreciate that. Gentlemen, you'll find that this subcommittee, I would say like most of the subcommittees, not all on the Appropriations Committee, we work well together, whether it's with our national labs. Mr. Foster and I, we have debates about other things, but when it comes to national labs, and he has a distinct scientific edge over me, but I still root for ORNL. But it is truly wonderful, the job that you are doing, and I think I can speak for the full subcommittee that we are pleased to fund this research. And I'm really thoroughly impressed by the discourse today. It seems that there's a wide variety, a wide spectrum of maladies and conditions that we're looking at. And the question that I want to ask, and it's a little bit broad, but I'm interested in all three of your perspectives. It seems as we fund this, and Dr. Varmus, you alluded to it with your earlier research, the genetic basis for cancer, particularly breast cancer. My mother passed of breast cancer when I was very, very young, and I'm so proud of the strides that we've made to combat that horrific disease. But my general question is for each and one of you all, is it genes? Is it environment? Or does it depend on the malady or condition? And how will our funding help narrow those questions and those gaps?
Just briefly, it is, of course, both contributions both of environments and behaviors as well as genetic inheritance. And it does, as you point out, vary from disease to disease. Right now, in the case of the neurological diseases we've been talking about, we probably know more about the impact of behaviors on how those diseases are treated, diagnosed, and evaluated. In the case of cancer, the one place where the Genome Project has made a major contribution that probably isn't as well appreciated by the public as it ought to be, to go back to your remarks, Madam Chairwoman. But we know that diagnoses are based on saying, this is a patient with breast cancer who has mutations in the PI3 kinase gene and the P53 gene. That makes a difference in deciding what therapies to use, because we now have drugs that inhibit the mutant forms of the PI3 kinase protein. So that is a major advance, and we understand some of the new immunotherapies based on what we know is happening in the genome. Those mutations are in general, not exclusively, but in general, not inherited mutations. Those are changes that have occurred because either of the randomness of the mutational process, or in the case of many diseases, by the exposure to certain mutagenic agents. For example, the agents that you inhale when you smoke a cigarette. We know what kinds of changes occur. We can look at the DNA of a patient's cancer and say, this patient was probably exposed to tobacco products, because we can see the signature of those products by our reading of the entire genome present in an individual's cancer. So the link between genetics and environment becomes very profound, and there's no longer a dichotomy between saying, I believe in the environmental causes of cancer and I believe in the genetic causes. They are merged in the action of environmental agents on our genome.
Thank you. Gentlemen?
So yeah, the answer is your genes or environment can help with your disease progression, or they can hurt with your disease progression, and we're learning a little bit about how those are risk, plus or minus risk factors, if you will. But, you know, I don't want to offend Dr. Varmus. It would be bad for my career. But we made progress
no
in
influence over your
cancer.
career.
I think we made progress in cancer a little bit because cancer is almost like a foreign thing. And if we just got rid of it, you know, then that was sort of the conception is that it would help. But the brain, you know, it's broken in a way and has to be fixed. He doesn't agree with me, but that's okay. That's okay. That's okay. But the regulation of the cells and the control is directed by genes and environment, and we need to get back to we need to know what a normal set point looks like, so then we can know if we've gotten back to it when we get an intervention.
Thank you.
Sounds like it's lose-lose, whoever I agree with for me. So but I'll try my own version, somewhere in the middle. I think this idea of genes versus environment when it comes to brains is as old as maybe as philosophy. Before we knew about genes, it was nature versus nurture, and after the discovery of it. And I think it's a very useful exercise to go back and forth to think it's nature or to think it's nurture. But in reality, it's probably some complicated loop where nature informs nurture and nurture informs nature. And the problem that biologists have, at least I have, when there's a loop, is that it's very hard to describe what causes what in a loop. If A and B are in a loop, did A cause B? Well, B also caused A. And I think that will only continue to happen, this sort of loop idea and how we talk about causality, the more we appreciate the complicated nature of a complicated organ like the brain. Did I do a good job in between?
Well, I think we're all in between.
Yeah.
Thank you, yes.
Where in the spectrum?
And I want to thank the gentleman, and I think that's great sometimes that we can have that difference of opinion. I think that's how we move forward. I think sometimes this body would learn from it as well, but I thank you, and I yield back. Yes.
Congresswoman Wasserman Schultz.
Thank you, Madam Chair. I'll just ask the committee's indulgence because this topic is a tiny bit personal for me. Some of you may know that I'm a breast cancer survivor. In December, I celebrated my 12th year as a survivor. I was 41 years old when I was diagnosed, and not to really get deep in the weeds on what I went through. When I was diagnosed, it was because I found a lump in my breast just after about 3 months after my first mammogram when I turned 40 years old. And it was kind of miraculous that I found it because it was less than half a centimeter, and my mammogram had been clean, so there was no reason to suspect that I needed to be looking for anything. I was very fortunate. So I didn't find my lump out of luck, I always say. I found it because of the knowledge and awareness that I had that I needed to pay attention to my breast health. However, and thankfully I was diagnosed at stage 1A, very early breast cancer. But subsequently, when I went through all the processes that you go through when you have a breast cancer diagnosis or any cancer diagnosis, is when I sat with the nurse educator and we went through the whole family tree process of the cancer in my family, and I had a lot of cancer in my family, but not really breast cancer. She suggested that because I was young and because I'm an Ashkenazi Jew, a Jewish person of Eastern European descent, for those of you that don't know what that is, and the amount of cancer I had in both sides of my family, that I may want to have a genetic test. So then she proceeded to tell me that as an Ashkenazi Jewish woman, I was 5 times more likely to have the BRCA1 or BRCA2 genetic mutation, and that gave me an 85% chance of having breast and ovarian cancer and smaller chances of having other kinds of cancer over the course of my lifetime, but particularly before I was 50 years old. Since I already had breast cancer, and I had been a state legislator, was immersed in the fight against breast cancer, passed breast cancer legislation in my state legislature, I thought I was pretty knowledgeable about my risk, about people's risk. You're 1 out of 8 women in our country will get breast cancer at some point in their lifetime. But I didn't know that as an Ashkenazi Jewish woman, I was that much more likely to carry the gene. And I had the test, went through the process of genetic counseling and all of that, and about a month later got the results. And when I opened the door to walk into the doctor's office, there were way too many people in the room for the result to be negative, so I didn't really have to open the folder. I knew that I was BRCA positive. And I have the BRCA2 genetic mutation. My question is focused on this. It can be challenging for most people to wrap their minds around the notion of biomedical research and development, and people's eyes glaze over when you're talking about oncogenes. And, for me and so many others around the world, the work that DOE and NIH do together and separately is very personal. And I always say when I tell my story that if I didn't know as much as I knew about breast cancer, about my risk, and that awareness, and we've had a lot of awareness that we've all focused on, attending so many different breast cancer functions and events. We have an entire month that's dedicated to it. We have a color that's been completely identified with breast cancer. But if I didn't know about my risk, then, and I can tell you countless women at risk don't know. So can you, all of you, explain how DOE and NIH projects like the Human Genome Project, for example, and subsequent projects, not only lead to the genetic tests that ultimately allowed me to be able to be more aware of my risk. The reason it was so important that I be aware of that risk and know that I was diagnosed with that gene, because my diagnosis at stage 1A, my initial course of treatment was to have a lumpectomy and radiation, and then I'd be done. But because I was able to have that genetic test and knew at how much higher risk I was, I had a double mastectomy. I had 7 surgeries over the course of a year. I had a prophylactic oophorectomy, meaning I had my ovaries removed at 41 years old. I mean, thank God I had all the kids we had planned to have. But I likely, if I had gone with the prescribed course of treatment and not been able to get that genetic test, I probably would have been dead, or I would have been diagnosed at a much later stage. So actualizing in a personal and simple way the incredible result of the research we've been able to do and the unlocking of the human genome. Can you talk a little bit about how we can make sure that we have wide public benefit and awareness of the development and incentivization of the technologies that you research, while also making sure that scientific discovery isn't stifled? You have a lot of work that you're doing that has little practical, might seem like it has little practical import, but can later lead to world-changing technologies, which has resulted in me being able to sit here to ask you this question. Mm-hmm. So thank you, Madam Chair, and I know I went over in my asking of my question, and I hope you understand my question.
Well, I'm not sure I completely understand the question.
The question really is very simply, a lot of this is mumbo jumbo to most people.
Yes.
And we spend billions of dollars on this research. It's critically important. But the awareness of the BRCA1 and BRCA2 genetic mutation was out there for many, many years. And someone as aware as I was had no idea about my potential.
No, you
risk.
you tell a story very powerfully, and I sympathize with the things you've had to go through. Getting information of this kind, which is complicated, to the public in an efficient and useful manner is difficult. I know from my experience at the NCI and NIH generally, and from working, I was the president of Memorial Sloan Kettering Cancer Center for 10 years, and I know that getting these messages out in a digestible fashion is a problem. One ancillary problem that's not widely appreciated is the scarcity of genetic counselors who specialize in this information. We have genetic counselors who are very abundant in advising you about your pregnancy, but we have not trained a cadre of similar counselors who can provide not just you once you've had your diagnosis, but people who are unaware of the need of what's entailed in having genetic counseling. The notion of...
Can I just interrupt? I'm sorry,
yeah, sure.
Madam Chair. For example, all 3 of my children are now at risk of carrying the genetic
Absolutely.
mutation. My twins are 20 years old. My youngest daughter is 16. Right. And my oldest daughter has said, well, I'm just going to go right away to 23andMe And, and 23andMe now can tell you whether you carry that genetic
course they
mutation. But having that information without that genetic advice,
Yes,
that
that's a big problem.
is really, I mean, to find that out in your kitchen table.
As you've
with
pointed
the
out,
entire enormity of what that means is, we can't allow that to happen.
it's a totally different question when you've had your children and you're willing to it's your attitude toward the surgical
Yes.
reduction in risk, which that's what you did. You reduced your risk of
Exactly.
breast and ovarian cancer by nearly
And
the
if I were
entirety.
younger, getting
that's right.
guidance on what, should I have my eggs extracted?
Right.
And
So
should I?
these are the difficulties created by knowledge that we are glad we have, because facing facts is what we want to do as a society. But the burdens that we create are significant, and we don't have clear answers. And we like to say that these are decisions that patients have to make for themselves, but they need guidance because the issues are complicated. The risks are not 100%, as you pointed out. They're high, and they vary from individual to individual depending upon the mutation that you found inherited. Now, you may have I'm 100% Ashkenazi, so I know the story. And if you have the mutations that are characteristic of people of Ashkenazi Jewish descent, we probably know a lot about those mutations. But there are also individuals who have fairly novel mutations in these genes. They're discovered when companies like 23andMe or your local genome center, as we have in New York, discovers the mutations. Then maybe very little known about them because every mutation carries a different risk. So these are subtleties that we're beginning to appreciate because of the vast amount of work that's being done by DOE and by NIH to look at as many genomes as possible. At the New York Genome Center, where I also work, we're particularly concerned about minority populations. Ashkenazi Jews are also a minority, but there are other minority populations that have not received the same level of scrutiny. And discovering the variants that are inherited that may predispose to risk of cancer or neurological diseases or others will only be appreciated if we include everybody in these genetic analyses that do predict risk of very serious diseases.
Thank you. And Madam Chair, the investments and I raise this because the investments that we have to make beyond just in this research so that we can have it result in these discoveries, help women like me and others and minorities that haven't had the application of these technologies applied just yet. I mean, the decision I had to make was I could go with the course of treatment based on the diagnosis that I had. But if I did that, the decision I had to make is have an MRI every 6 months for the rest of my life and constantly waiting for the other shoe to drop. I was at a stage in my life where, thankfully, I got married young. We had our children. I had breastfed my children. Those were all decisions that I had to make in a very short period of time because I had cancer. And we need to make sure that there's a lot of results and impact that we have from the discoveries that we make that affect real people. And we need to be making investments as appropriators to try to make sure that they can deal with the ramifications of the results of these incredible discoveries. So I appreciate you indulging me for way over my time. I yield back.
Could I make a brief comment, Chairwoman Kaptur?
Dr. Chance?
Mrs. Wasserman-Schultz, thank you so much for sharing your story. I think the willingness of people to share stories and patient advocacy has been so important to moving the needle and getting it forward. And I want to echo Dr. Varmus's comments. We're discovering this stuff, but if there aren't people in the medical system who can explain it to you, right, patients won't get the benefit. And genetic counseling, we have genetic counseling training programs at CASE and around the country, but often reimbursement is a problem. Genetic counseling is not necessarily reimbursable in many plans, and as more complicated genetic information comes in, it's going to be a little more expensive and a little more challenging, to bring that information to patients. But those are the professionals in the healthcare system who could really help us.
Well, I just want to say to Congresswoman Wasserman-Schultz, you could have as much time as you want. And we admire you very much for your courage and your continued service. Thank you so much. Congressman Newhouse.
Thank you, Madam Chair, and Mr. Simpson for having this hearing today. It's truly an important subject to discuss. I want to thank Congressman Wasserman Schultz for her sharing her testimony today. It helps us understand the significance of what you're working on. And I won't extrapolate a lot, but this kind of work impacts almost everybody. And let me tell you, as someone who held the hand of my first wife as she fought breast cancer, even though you are coming up with new things all the time, let me just tell you, it isn't fast enough when you're in the situation where a loved one is facing something like this. So anything we can do to accelerate your ability to come up with things not just in cancer, but in brain research and all the other disciplines that we're focused on. But so thank you, all of you, for your work. It's the Lord's work, and very much we appreciate as Mr. Fleshman said, we all support doing as much as we can to further that work. So I'm from the state of Washington. I represent among my constituents are those that work at the Pacific Northwest National Laboratory. Truly a tremendous institution that does a lot of work in so many different areas. Like Mr. Simpson, I'm always amazed at the things that they're working on there, and also, I hate to admit this, but like him, don't always understand what they're doing, but it's great stuff and making tremendous strides in much of their work. So I'm proud of that, and I always try to point out what I think is really one of those things that a lot of people, even in the Tri-Cities and the state of Washington, aren't aware that what's going on right down the road. And so thanks for giving me a soapbox to talk about that. But as much of the partnership between the NIH and the DOE, as you've talked about, Dr. Varmus, it's been focusing on the world's computing systems, and I think the fastest one happens to be at Oak Ridge, if I'm not mistaken.
And the next fastest one coming.
Oh, is that right? Okay. But you also talked about other things that DOE and the labs bring to the table. My understanding is that several of the NIH programs look to PNNL for things such as the advanced chemical analysis systems that can be very crucial for understanding the impacts of, say, exercise on our health. And even like Ms. Wasserman Schultz was talking about, helping do early detection of cancer and potential treatments. So I guess that struck me as being something. What are some of the areas beyond computing that you see some of the DOE facilities and the researchers having an impact on things like cancer, or where should we be encouraging that into the future?
You're correct, Mr. Newhouse, that all of us have been emphasizing to a very large extent, but not exclusively, the computational power that DOE brings to the table when DOE and NIH undertake things together. Indeed, the cancer initiative I described is largely based on the interface between high-performance computing and certain areas of cancer research. But as we heard, and as is manifest frequently, but as we heard during our workshops in putting together a report on NIH-DOE interactions, there are many other areas: materials science, developing new ways to fabricate things; nanotechnology, the creation of very small devices that can be used, for example, in drug delivery or in delivery of radioisotopes. Those are areas of research where NIH and DOE are doing things that are similar and benefit from the interaction. You talked about sensors, and sensors can range from sensing exercise regimes to sensing molecules in the bloodstream. Please, Mr. Simpson, who alluded, or was it Ms. Captur, alluded to devices for detecting head injury. We can now measure deep DNA that comes from cancer cells in blood by taking a peripheral blood sample and use that as a guide for whether treatment's actually working. That's a kind of sensor developed largely through NCI and the private sector in this case, but representing the kind of sensitive device for finding just individual small numbers of molecules that can reveal the function of an important organ or the progress of disease. So these are all areas, and mainly coming from engineering and the physical sciences, which has been the strong point for much of DOE-related research, but as I tried to emphasize, has historically been involved in many of the advances we've made in healthcare, from the earliest, the development of simple X-rays by William Rankin over 100 years ago to more modern ways to use magnetic resonance imaging or PET or CAT scanning to diagnose disease early. All these represent efforts to understand the body not just as a living set of cells, but also as a physical entity made up of molecules that are sometimes best approached through physics, chemistry, and engineering.
I have perhaps a tangential answer. So about 2, maybe 3 years ago, I never really heard of the National Lab System. I was 100% a neuroscientist. I maybe heard of some national labs, but certainly not of the 17 that exist. So when I first showed up, I was amazed by the technology, the X-rays, the computing, the material sciences. But I'd like to add one other thing, which still surprises me, and I think Dr. Varmus alluded to it earlier. There's a culture about the National Lab System that doesn't exist or exists at a less prominence in academia, and that is that impossible ideas seem common in the National Lab System, and people there are willing to address things that have a scientific basis and a physical basis, but in other settings, the scale is beyond what people could imagine. And I think this culture, this part of that National Lab System is crucial and valuable. It's a place where impossible ideas are routinely considered, and that, or what are at the moment considered impossible ideas are routinely considered. And there's a different risk versus reward analysis that's happening, I think, in the national lab, in the DOE system, that's different for appropriate reasons in the NIH world, where the risk versus reward is calculated differently. And I think combining these cultures is going to actually help advance science, even if there were no X-ray sources or computational sources or material sciences sources.
Maybe I'm way over time, but that may give me an opening to ask my second question. Well, if I were one of your students, I'd say Dr. Bobby, but Dr. Kasturi? Yeah, okay. So I was interested in your comments, and I wanted to ask you if you were familiar with the Allen Institute in the state of Washington, their efforts on brain science. That I've just learned a little bit about some of their things that they're working on, and like the Brain Initiative and the potential partnerships with the NIH and DOE to facilitate further scientific discoveries. They've been successful on large scale transformative neuroscience projects, including just what you mentioned, the mouse connectome, as you referred to, you talked about that, but also wanted to flag that the Institute does provide open access of the data for scientists worldwide, free of charge, yes. And which has partially been made possible by the congressional funding of the Brain Initiative. So should also point out that the Institute has a history of forging collaborative partnerships with other DOE centers of excellence involving neuroscience projects, such as the Big Neuron Data Analysis Hackathon at Oak Ridge. And I know a lot of the great folks at the Institute look forward to more opportunities for comparable partnerships with those initiatives. So just wanted to bring that up. Any thoughts you might have on that as well?
I should say that the Allen Institute for Brain Science has some of the premier scientists in the world studying brain mapping, the way we do it. We are at worst frenemies, at best very close colleagues. I cannot imagine a kind of effort at a large scale doing this kind of mapping, not including places like the Allen Institute or Genelia Farms, as Dr. Varmus mentioned earlier. These people have great experience in how to do this. And I think it's this combination of private institutions like the Allen Institute and Genelia Farms mixing with federal institutions where we will have really great breakthroughs. But in general, both the data they collect and the way they distribute the data for free to the community is a kind of model to emulate.
Yeah, good. Thank you very much. And again, thank you all for being here. Dr. Chance as well. Appreciate your testimony. Thank you, Madam Chair.
Thank you, Congressman Newhouse. I wanted to ask the Mouse Connectome effort, is that finished?
No, I wish. I think we are just about to start that effort.
Where?
Not well known where or how, but I know, in fact, I was just speaking with Dr. Varmus about it this morning. It is an effort that is just literally starting, that people are thinking about how to fund it, what are the various multi-agencies that should be involved in it, where are the various places that it should be developed and instituted. But I think the effort is actually at the very beginning as opposed to even close to the end.
So it involves several government instrumentalities and several private sector instrumentalities?
Absolutely. It will, I suspect, involve. So, for example, places like the Allen Institute, as Representative Newhouse was saying, places like Genelia Farms, which is a Howard Hughes private institute, like Professor Varmus was talking about, in addition to government institutions, DOE labs, and a bunch of academic places together, I think, will be how the connectome gets mapped, if the genome is any example, which was also a very similar process of mixing private, public, federal, and academic all together.
Right. I think, I'll speak very openly, I think science has trouble communicating with the general public. And in order to ease that difficulty,
Yes.
Congresswoman Wasserman Schultz offered the observation about how she learned and
Yes.
what she knew and what she didn't know. I would urge you to consider somehow with you're scientists, you're not necessarily communicators, though you're the best communicators in your field or you wouldn't be here today. But if you were to look at various months or days that Congress designates for whether it's cancer,
Yes.
whether it's mental health awareness, and think a little bit about how you would use those opportunities, because many members are involved in trying to use those moments to help the people they represent and help the country. I don't think science uses those days very effectively. So, for example, on autism, and I honestly don't remember which day it is that we acknowledge the issue of autism and we try to draw attention to it. The cancer issue is another one. But at that moment, you could anticipate, you can get a calendar, yes, and you can look ahead, and let's say it's June 1st, right? At that moment, the best knowledge we have could come here to Washington. We could give you the Gold Room or the Rayburn Room, or we could find a room here, a big room, and all of the associations, the Alzheimer's Association, for example, or the National Cancer Society, whatever the individual concern is, we could use that moment, and you would have 436 members of the House plus all of our territories, and we could try to help message. But honestly, members are very busy, and they don't keep up on what's happening with the pace of science. So, for example, a member like myself, I think to myself, okay, so if mental illness is an issue I care deeply about, I know of the Stanley Foundation. But Dr. E. Fuller Torrey, in a lot of what I see on the website, is physical brain presentation. It isn't necessarily neurotransmitters. So the synapses in the middle part of the brain, right, are larger, doctor, right, according to at least his research. one of our esteemed congressmen from Ohio, Mr. Gonzalez, is a former football player and is very involved with Boston University and the brain bank they have there. So I go to the website and I look at that, trying to help him achieve what his goal is, which is healing and understanding. And I'm going, well, is it more physical, or is it more embedded in the parts that we can't see? It's really difficult to be a member and try to figure out, okay, so where should I be moving this vast ship? And I'm even having a little difficulty this morning trying to figure out, respectful of what our jurisdiction is, okay, so do we need more advanced light sources? Do we need more larger computers? How far along are we on that road? It's hard. I'm reading, Dr. Varmus, what you submitted to the record, 1, 2, 3, 4. Do I believe this is all going on? And it's already happening, so we don't have to worry about it? But it seems like on the communications front, we're not effectively using science to help the American people move forward in this arena. We could. NIH and DOE could think about that, and maybe in 2020 pick one. It could be cancer, it could be autism, it could be bipolar. Whatever it is, how effective could we be in reaching the American public? And at that moment, the best scientists we have, the best spokespeople we have could come forward, deliver some papers, talk about the road forward, 1, 2, 3, 4. All I know about autism is I looked through a microscope in one lab, and it showed that the receptors inside the ear, rather than looking like this for sounds and so forth, look like this. They're all blunted. I thought, okay, well, what does that mean? Every member has that kind of experience. They can't step away from the particular and kind of figure out, all right, so what do we say to the American people? What do we know and what don't we know? What are these murky areas? And what we know, maybe there are certain things we know. We know that for many neurological conditions, they onset in the teens or something. What do school teachers look for? What are they? Many scientists probably know, but we don't communicate it very well. So I'm just saying to you, maybe if you could draw from your work in cancer, your work with light imaging, wouldn't it be great to be able to tell the American people, well, we have to have a machine 10 times more powerful than this, or we need more computer scientists that will major in whatever to solve this. I don't think we pick the essential out of what we already know and help direct thinking of the public and members as a result. Dr. Varmus?
Thank you for bringing up this important issue. I think many scientists, myself included, have been worried about this for a very long time. For over 25 years, I've been affiliated, for example, with the Congressional Biomedical Research Caucus. And as you point out, although not just one entity, it's a lot of different sciences. You heard that around, even on this small table. What do people want to hear? I think one of the things that I resonated with in your comments just now was the idea that a committee like yours can distill a specific question. That is, what is needed specifically to promote effective and more productive research between the two agencies? We have this report. It covers a lot of things. There's now 4 years of history since our report was issued. One could imagine having even just a discussion, not even a hearing, in which you say, all right, what do you think are the weaknesses? Where can we put our resources to make these collaborations more productive? You know, there's so many things out there, and you heard a lot of ideas just during our discussion this morning. But I think the pointed question from appropriators themselves could be very beneficial. I think a lot of us actually have spent a lot of time trying to communicate with the public. The public is a large, diverse beast with a lot of different levels of competence, and there's a lot to say. and being told by you, the appropriators, what specific questions you'd like answered, I think is something that would go a long way to improving the interchange between scientists and you as representatives of the public.
Would a discussion, Dr. Varmus, more of an informal discussion between DOE and NIH be a valuable, taking your recommendations from the hearing this morning, would that be valuable?
I think that is actually, the discussions between leaders and the agencies is already occurring at a very high level. I was very gratified in my conversations with Chris Fall, the head of the Office of Science, with Francis Collins, and with others at NIH who are running these collaborative efforts. There are a lot of discussions occurring. I think what is maybe missing in the mix is having interested staff, especially from the appropriation side, say, we'd like to hear from both of you in the room, so what can we as appropriators do to make these connections work better? Right now, people are trying to make better use of the money they have and put things together in a sensible way. But I think actually talking to people who control the budgets would be a very useful exercise.
All right. Yes, Dr. Chance?
Thank you, Chairwoman Kaptur. I really accept a lot of what you say. I accept from Mr. Fleischman, we're not moving fast enough. I accept your admonition, we don't communicate well enough. And I accept your challenge to come and make that case and get involved and do more myself. Further, I think if we want to go faster, the beam lines are not fully, utilized. There's spare capacity, and we could go faster, and maybe that would be a cost-efficient way. New beam lines take a while, and so those can't help immediately, but we could get more scientists embedded with the DOE. I mean, that is my secret sauce of what we've been able to do. Take university scientists and embed them with the DOE scientists. And so those stakeholders are bringing talent, money, and all of their resources to bear to sit on that baseline which you so generously appropriate. Thank you.
Dr. Chance, could you just, thank you for that statement. Could you write me back for the DNA samples that I believe rest somewhere at Case?
I know quite a bit about that project. We work very closely with our Veterans Affairs Hospital. They're a key affiliate. I know Dr. Bonomo, who's on some of those samples, and I will investigate that for you right away.
Why are they useful? I have no idea. Shouldn't I?
Yes, you should.
So it's an unknown, big question mark. The Boston University Brain Bank, is that valuable? Dr. Keshuri?
So we are collaborating with them now to try to take some of these samples from the Boston University Brain Bank and analyze them. Sorry, these are samples for post-mortem samples of people who have either been diagnosed with concussive neuropathies or et cetera, and age-matched controls, if you will. So we're working with them right now to see if we can use our microscopes to find the physical basis of these different diseases. It's just an exploratory work now, but I find these databases and tissue banks extremely valuable because when they were collected, they were probably collected for a particular reason. But as our technologies advance, as our microscopes get better, as our genetic analyses gets better, these tissue banks become super valuable for new questions that had never been emerged. When they collected the tissue at Boston University, they didn't collect it to have someone do super-powered nanoscale electron microscopic imaging of it. But it turns out that we're trying that now. And it's a resource being applied to a new question that no one had ever thought of, which I really like in this world.
And I know our ranking member obviously talked with Secretary Perry about resources in California. I don't know exactly what exists there, but you probably know.
Yes.
And there's interest among members not on this committee. That's a good thing.
Yes.
We need to use that. I just wanted to say Dr. Varmus, I think, has to leave quickly because of a travel arrangement. Is that correct, Doctor?
I have to teach at 5 o'clock in New York. So I need to get on the 1 o'clock Sala.
Yeah, boy.
I'll make it.
All right. Well, I just wanted to thank you all very much on behalf of our committee. We had a great turnout this morning. That shows the deep interest. I hope that as you go back to your respective responsibilities, that you will consider the questions this morning and the possibilities that exist with the additional funding that the ranking member and I tried very hard to get in this recent bill, and how we might move some of the investments at the Department of Energy forward, whether it's in light imaging, whatever you tell us, to help you to move your research forward faster in collaboration between agencies and departments and outside interests. I hope that you use this as an open door, and I took your recommendation, Dr. Varmus, and we will try to bring, with your help, together some of those who should be around the same table as we decide what is an appropriate role for the Department of Energy, what is an appropriate role for NIH, what is an appropriate role, frankly, for defense medicine, and for the research that occurs at the Department of Veterans Affairs. I don't view that research as everybody being in their own car, for heaven's sake, but we need to be on the same train car. We need to be working together here, and we have to break down some of the stovepipes that inhibit faster progress on scientific inquiry. So consider this, please think about what you've heard others say and ask this morning, and give us your thoughts. We greatly appreciate your testimony this morning and your time. Mr. Simpson, do you have any additional comments or questions?
No, you better go if you're going to make the 1 o'clock.
All right, very good. That concludes this morning's hearing. Again, I would like to thank our witnesses for joining us today, and I ask them to please ensure for the hearing record that questions for the record and any supporting information requested by the subcommittee are delivered in final form to us no later than 3 weeks. From the time you receive them, members who have additional questions for the record will have until the close of business on Friday to provide them to the subcommittee office. This hearing, with our gratitude, is adjourned.
All right. That was fun, wasn't it? Okay, let me go ahead and pause this before they try to autoplay something else that's copywritten or something. Thank you guys for joining. Thank you for joining the Red Sonja. Yep. Or something real scientific. Yes, exactly. That's our government for you, right? They're just absolutely wonderful at working together. I thought it was interesting how the representative at the end was saying, I think the scientific community isn't good about communicating to the public. Well, scientists in general, oftentimes, now I'm a nerd, so I feel like I can say this, okay? I'm a total nerd, I'm a total science geek, I could talk quantum physics all day long, and molecular biology and all kinds of stuff. But it seems to me like they are putting the emphasis on that scientists need to be the ones talking to people, and she did bring up the fact that scientists are not generally that great at, communicating with the public. A lot of people that are nerdy or scientific and stuff, we like to sit in the background and research things. We don't necessarily like to be like how I'm doing right now. Like this. I'm not all that great at this. There are people that are way better at this than I am, but they don't, they might not know the science behind it. So just because you know the science doesn't mean you're going to be great at communicating it to somebody else. So I feel like they really need to take some ownership with that. If the scientific community is not great about communicating with the public, then you need to have a public relations officer for the scientific community. if you don't know, sheriff's departments, police departments, and stuff like that, they have a specially designated information officer. And that person is the person that deals with the media. They are the ones that make the social media posts. They're the ones that schedule conferences and hearings and all this other kind of stuff. They're the ones that get out there. And if you've ever seen some funny posts from police departments and stuff like that on Twitter, that's the public information officers. That's what their job is. They are people that are supposed to be good at public relations. And it seems to me that with this enormous emphasis that we have in society nowadays on talking to people through social media, and there's influencers and stuff like that. Well, people that are really big online and that have a whole bunch of followers and are really good at just talking to people and just drawing people's attention, they're not necessarily going to want to just sit around making Instagram posts the whole time. So how about you actually try to draw in some people like that into the political or government, I guess, job role, where they would be able to do pretty much that same thing, where they're communicating with the public and they're talking about things in an easy to understand manner and everything like that. Why not draw them in there? They're already good at that. There's a lot of people that are trying to do that stuff. There are people that want to all they want to do is to be able to talk to other people. So why don't they have people specially designated for that? Arg. This really upsets me when I hear them making excuses, in my opinion, for medical information and scientific information not being out there. Part of the problem that I think that they face is that scientists expect the public to have the same awareness and the same understanding of at least some simple biology or simple chemistry or physics. And the problem is that the public doesn't. That's not their fault. That is the fault of our education system. So our education system is controlled by the government. It's not educating people properly on the harms of what they're putting into their bodies. It's not even educating them on what actual proper nutrition is, on how to stay healthy. None of that stuff. I never even had to take a birds and the bees class in school. I went to a private school, the most prestigious private academy in the state that I was in. And I never had to take a birds and the bees class. Never. I had to take some other chemistry and some other physics, though, that other people don't have to take. So how does that make any sense, Oh my goodness. And then the doctors, not even the doctors, are required to take long, extensive, nutritional classes and courses. So that's why you see fat doctors giving diabetic patients advice on what to eat. And it's what? Why would they listen to you? They're not going to listen to you. My internet's going crazy now because it's storming. But I really get upset by this kind of stuff. I really do. I have a lot of friends and family members that are greatly affected by medical issues. I'm affected by medical issues myself. A lot. Part of the reason why I switched my name from being something that implied daily stuff to something that doesn't is because sometimes I can't work. I can't do my normal stuff. So for them to just, be oh, this excuse and that excuse and, this equipment is so costly and these tests are so costly. You're the ones that have been allowing the pollution in our country. You're the ones that have been allowing all this stuff. They give out warnings to companies to stop promoting their material in a certain way. They give out a warning. The FDA issues a warning to Purell to stop saying that their hand sanitizer kills things like the influenza and Ebola. Stop issuing them warnings and tell them that they need to shut the F down until they stop. And then, but the government can't have that much control. They're the ones that didn't do their job in the first place. That's why you're being harmed by a corporation. Don't take the side of the corporation that, we can't have this big greedy corporation closed down. Oh no. The government should have not allowed that in the first place. And since they have, then when all these extra costs come up, then who should pay for it? I don't know, maybe the government that allowed it. Stop giving them these huge salaries where they are able to have multiple cars and go on family vacations all the time, while we have people in our country that are dying because they can't receive the medical care that they need. Like, are you kidding me? Are you kidding me? It really, really frustrates me. The doctors, they act like, oh, well, doctors don't know about this stuff. Doctors get sent notices by the medical community, by NIH, by all of these different places. Whenever there is new medical science that comes out, they get notified. Doctors are supposed to go and get up-to-date training. They're supposed to be aware of the new stuff that is found and coming out. If they are not, that is their fault, and they should not be a doctor. That's how it works, okay? Like, stop allowing these greedy doctors that are just prescribing their patients opioids instead of telling them how they can fix their problem. They should not be doctors. And the government is just allowing this, and then they want to go, oh, well, we need to take more tax money from people so that we can spend more money on this medical thing and that medical thing and, scientific advancements and everything. How about you just go back to the companies that you allowed do this in the first place and you take the money from them? Because they're all run by billionaires, okay? So they have the money. Go take it from them. Or how about you dish it out of your own pockets? Seems to me that should be one of the two options. And if you didn't want it to come down to that ultimatum, then they shouldn't have allowed it in the first place. All right. So now that I'm done ranting with that, by the way, welcome to Grow the Info. I don't usually rant like that. I'm sorry. This is a situation and something that is near and dear to my heart. And I'm tired of seeing my friends and family being in pain and, some of them being on that last leg of life because of completely preventable things that our government just wants to totally wash their hands of. And they're still dragging their feet. This is how the two-party system is just a joke, because it's not fixed, is it? None of it is fixed. It doesn't matter who gets into office. It doesn't matter who takes over. It doesn't matter if it's Republican or Democrat. It never gets fixed. They sit here and they talk back and forth about it nonstop, and oh, we should do this, we should do that, and they drag their feet until it's too late. Speaking of which, I gotta stop this stream because there is a hearing on the coronavirus response by the U.S. government that is about to start soon. So make sure that you're subscribed and turn on bell notifications so you don't miss that. Again, I'm sorry that I was ranty. This stuff makes me so mad. Thank you guys so much for joining, though. I really hope that the stream works. If it doesn't work, I don't know. I hope it does. Alright, I'll see you guys in a little bit. And remember, you can follow us on Twitter and Instagram at GrowTheInfo is our handle. And you can check out the website growtheinfo.com for resources and information, press releases, all kinds of good stuff. Thank you guys so much for joining. Have a wonderful day, and I hope I will see you here in a little bit.